All right, AMP. So what we're going to start talking about now is a little bit more about really how we start sending these impulses. First thing I want to talk about just a little bit of the history about how people figured out how this was working. It was really a gentleman by the name of Otto Lowy that came up with knowing that these were actually the nervous system was actually neurotransmitters and chemicals and not electrical. People have known for a period of time that obviously there were some electrical connections to this, in the fact that we can electrocute a muscle and cause a contraction of it. But we knew there was some electrical activity happening with the body. We just weren't sure if these synapses and how we were sending all this communication through the nervous system was electrical or chemical, at least especially in between. So some of the kind of ideas that were leading us to believe that it was not electrical transmission and that there was actual chemicals at the synapse was really the fact that there was always a delay if you actually did a voltmeters on both sides. And they started figuring this out in the 18th and 19th centuries. So in the 1900s there for the most part. But that delay is actually, if it was electrical, this is too long for that to be taking place there. And also, if it was all electrical, you would think that if you could stimulate on the other side of the neuron that was downstream, that you could get a response in the presynaptic one. It's kind of like if you're sending electricity, it doesn't matter where you put it on the wire, it's going to travel the entire wire. The other thing is that a lot of times if you stimulated a presynaptic neuron, you would get sometimes inhibition on the other side, which that gets difficult to explain with just electrical activity. And then finally, there was never really any relationship between the magnitude of the presynaptic and postsynaptic events here. So there was, like, how big of a stimulus presynaptic didn't really change what was going on postsynaptically. So how this was kind of determined was actually Otto Lowy, like I said, in 1921. And what he did is, they had known that if you electrically stimulated the vagus nerve on the heart, it would actually slow down the heartbeat. So what he did on this one is, you could put this frog's heart in this ringer solution, which could keep the heart alive. They would stimulate the vagus nerve on this donor heart here. And what they had going on here is they had a solution that was going into that heart. They then had basically a tube that was taking the fluid from the one heart and bringing it down to a second heart. And what they saw, if they stimulated the vagus nerve on that donor heart, they would also get a change or decreasing of the heart rate downstream in this recipient heart, where there was definitely no electrical connection between this donor heart and the recipient heart down here. What they actually he called it vagus, so the vagus nerve. But what it actually was it was the neurotransmitter acetylcholine was being moved from one to the other. And this was really the experiment that kind of showed that these synapses were actually working in a chemical fashion. And again, these synapses are where this connection takes place, and really where we can take information from one neuron and get it to the next one. And there is actually some electrical synapses, they're just very few and far between. For the most part, these are chemical synapses. And again, like I said, the chemical synapse is the most common one here. You have the presynaptic neuron that is going to produce a chemical signal that's going to go into that synapse and move across and bind to receptors on the postsynaptic neuron. This is generally going to be a dendrite of the next neuron. And again, it's coming up really, really close in just a small gap. So if this is synaptic end bulb and this is the next dendrite, it's going to come up really, really close. And then, again, that chemical is going to move across, or that neurotransmitter is going to move across that gap and bind receptors on the postsynaptic neuron. Again, release from that synaptic knob, they diffuse in there, move away, move across. There is a little delay in the time between that release and when it binds. And the other thing is that postsynaptic neuron, this allows processing to take place a lot more, in that you can have multiple neurons affecting that postsynaptic neuron. So why we might be talking about this one particular synapse, there's going to be other synapses on these dendrites that can either stimulate or inhibit this neuron to determine and allow it to do processing, for example. Electrical synapses, they do exist. They're just very, very rare. And these ones are where they're actually technically touching, and there is actual gap junctions that will let those ions move from one cell to the next. Again, certain areas of the brain, certain things with the eyes that we do see this in. But again, not going to spend much time talking about it because it is very rare. When we start talking about how we label some of this stuff and how we name it, what you're going to see is groups of cell bodies in the central nervous system. We refer to this as a nuclei. So when we get to the brain in AMP2, we'll talk about the basal nuclei. This is a group of cell bodies at the base of the cerebrum. If we were talking about a group of cell bodies in the peripheral nervous system, this is going to be a ganglion or a ganglia. So the dorsal root ganglion is an example of that. When we talk about these nerve processes, which are generally going to be groups of axons together, in the central nervous system, we call this a tract. So like with the spinal cord, we'll talk about ascending or descending tracts, where if we're out in the peripheral nervous system, we refer to this as a nerve. And again, it's really these processes are going to be those dendrites and axons that are leaving that. Generally, when we're talking about tracts and nerves, we're really talking about these axons, or kind of whatever you want to call the one that is that continuous axon on them, for example, like a unipolar neuron. And these axons, we're going to see, they have their own little membrane. They call it the axolemma. It is surrounded in a lot of cases, not all cases, but in a good vast majority of these neurons, that axon is going to be wrapped with something called a myelin sheath. Myelin is a lipid, and this cell wraps around here, and this is going to electrically isolate this, kind of like plastic on a wire that makes it so this doesn't ground out. And what we're going to see later on is this allows us to do something called saltatory conduction, which is going to increase how fast this action potential can travel. What we're going to see that this wrapping doesn't ever completely cover it. There's always going to be little spots that are uncovered with no myelin. These are called nodes, referred to as the nodes of Ranvier. In the central nervous system, it would be the oligodendrocytes doing this. In the peripheral nervous system, it's going to be the Schwann cells that are allowing this to happen. But again, you can see here, this myelin wrapping, this would be Schwann cells, and you can see that would be the nodes of Ranvier in between. Like I said, this myelination is made by these different glial cells, has a lot of lipids that are important in it. There is some diseases where you don't make the myelin correctly. There was a really old movie called Lorenzo's Oil. Nick Nolte, Susan Sarandon, that was talking about this. Again, it is going to be those Schwann cells in the peripheral nervous system, those oligodendrocytes in the central nervous system that are doing this job. Really what happens here, at least in the peripheral nervous system, the neurolemmacyte's going to get on there and it's going to do these kind of continuous wrapping. So if this was like the nerve thing right here, it's going to wrap and twist up and around it like this and continue to make this. Again, the cytoplasm and the nucleus get pushed way out to the side here, and they call this little tube now that it's traveling through the neurolemma. So you can see an example of this right here. Here's the axon that Schwann cell is going to surround it, and then it wraps up tighter and tighter and tighter, pushing all the cytoplasm and the nucleus out to the outside here. And you can kind of see these in the picture right here. This stuff doesn't stain particularly well, so you can see the myelin sheath right through here. Lipids don't stain well, so you can see it lit up right here. That would be that myelin sheath with the axon traveling through the middle of it. So with neurolemmacytes or Schwann cells, they each do about 1 millimeter of an axon, so you're going to need hundreds of these to myelinate one of these long axons. A whole bunch of them here. And again, the spaces in between are those nodes of Ranvier. Oligodendrocytes, these ones, each little extension is going to do about 1 millimeter of these axons. Again, doesn't do that neurolemma. And with these oligodendrocytes, they a lot of times are going to be wrapping around multiple axons and not just 1 millimeter of one axon. So you can see, for example, this right here has 5 extensions that are all doing some wrapping of these axons in the central nervous system. There are some unmyelinated axons. A lot of times these are associated with these Schwann cells, but it does not have a myelin sheath that completely covers them. It's just kind of like, it's almost like somebody dug a trench in these cells and they're sitting down within this cell. Again, not wrapped in repeated layers. We don't see any unmyelinated axons with oligodendrocytes. So you can see something like this, where you get these Schwann cells and it's going to be kind of housed in there like it's a little trench, but again, exposed, not completely covered. And again, this is going to affect how these send impulses. These are going to be much slower than a myelinated axon, for example. And again, there is definitely diseases that cause issues with this. So multiple sclerosis, or MS, is a disease in the central nervous system where the immune system is attacking these oligodendrocytes, and it leads to damage of those. And again, when the cell signals can't be sent correctly, this is going to lead to problems. So MS, lots of ones here. I mean, it can be fatal over depending on what it goes to, but it can cause vision problems, muscle spasms, bladder problems. Definitely a lot of those. Guillain-Barré is a kind of similar thing, but in the peripheral nervous system. And this is usually due to an infection. You'll get inflammation, and usually this will resolve on its own, but can a lot of times cause some issues with muscle weakness when this is happening. What we're going to see is in the peripheral nervous system, you can also get regeneration of an axon. We do not see this in the central nervous system, but if you get an axon that's severed in the peripheral nervous system, it's going to seal off, and really the distal portion of it that's going to the target is going to degenerate. Wallerian degeneration, if you really want to put a term to it. I'm not expecting you to be able to go through all these steps, but just wanting you to know that you can regenerate a peripheral one here. But that neurolemma, those tube, kind of survives here, and that's going to help form this regeneration tube, which is going to let this travel back up through it. So we're going to get regeneration and then remyelination through this, and at some point, I mean, and in some cases this might be months or years for this to reconnect, depending on how long that axon needs to regenerate, but you will get innervation restored over a period of time in the peripheral nervous system. So you can kind of see right here, this is damaged. This end here is going to degenerate, but we keep that neurolemma here. We're going to get this regeneration tube. That axon is going to help be retargeted and again go back to where it was. Again, this is not particularly fast, so it will take a period of time for this to go back. It's not like you sever this and in a week you have this sensation or this movement back. It may take months or years in some cases. The central nervous system, it is very rare to see much of this happening here. The oligodendrocytes, really these molecules that don't let this happen. And there's lots of axons in there that tend to get filled in with astrocytes if there's damage to these ones. There has been some studies where they're using some nasal stem cells and certain growth factors that were leading to some limited reconnection of spinal cord injuries. So there are ways that we are medically starting to be able to do this a little bit, but really with normal body function, generally something that damages an axon in the central nervous system tends to be rather permanent. So that at least kind of gives you a little idea of what's going on with these synapses and the different types. What we're going to get into this next one here is really how we generate nerve impulses, and then we're going to start moving into how do we go about interpreting these nerve impulses? How do we send in information? So that is what we'll talk about next time.